So after the introduction of flow cytometry, now we will see how it could be applied on the plant cells by Sophie from University of Hasselt.
Okay, thank you. So Carla already gave an excellent introduction on flow cytometry, so I think I can really skip most of the basics. But today what I want to talk to you about is how we can use flow cytometry to analyze the cell cycle. Carla already mentioned that this is also one of the applications that you can use flow cytometry for. But we specifically do this in plants. So in my research, I'm working on the model plant Arabidopsis. So I will show you some of this application today. So what I would like to talk to you about today, short overview. So first of all, I will explain to you the differences between the classical cell cycle and endoreplication, which is an alternative version of the cell cycle. I will, of course, show you how you can measure endoreplication using flow cytometry. And then I will show you some examples of how this process is involved, first of all, in plant development, but also in responses of plants to different stress conditions. So we'll start first with the differences between the classical cell cycle and endoreplication. So I think this is something all of you know, but I will just briefly go through it again. So the classical cell cycle in animal cells, but also in plant cells, consists of 4 different phases. So first of all, we start off with the G1 or gap1 phase. Then we have the S phase, in which the DNA of the cell is replicated. Subsequently, there is another gap phase, and in these gap phases, cells actually prepare for the next phase. And then in the end, we have mitosis, where the cell will divide into 2 daughter cells. And for a typical human cell, one cell cycle takes around 1 day. And this interphase, which consists of gap1 as in gap2 phase, takes approximately 23 hours, whereas the M phase or the actual cell division only takes around 1 hour. Now here on the left, we will have another look at this classical cell cycle with the 4 phases I just mentioned. But what's very important now is that we will have a look at what happens to the DNA content of the cell during these different phases. So we start off here with a cell with a DNA content of 2C, where C represents the haploid DNA content. What will happen then in the S phase is, of course, the DNA is replicated, so it's doubled. So the cell doubles its DNA content to 4C. Nothing changes to this in the G2 phase, but then this 4C cell will divide into 2 daughter cells, and these, again, each contain a DNA content of 2C, which is the same as the original cell. And they can go through another cell cycle and so on. But in the end, the DNA content remains the same. Now, what happens during an endocycle or endoreplication is, again, we start off with a 2C cell, but then it will go through the S phase, but it will skip G2 and M phase. So after the S phase, it will go straight back to G1 phase. So what happens is that with every consecutive endocycle, the cell will double its DNA. So first we start with a 2C cell, then after one endocycle, the DNA content is 4C, after the next cycle, it's 8C, and so on. And in Arabidopsis, so the model organism, the model plant that I'm working with, these ploidy levels can reach up to 32C. And this process, or what it leads to, this enhanced DNA content, is what we call endopolyploidy. So for example, Arabidopsis, it's a diploid organism. It will stay a diploid organism, but some cells within this organism will have a higher DNA content, and that's what we call endopolyploidy. So as I mentioned, endoreplication is a modification of the classical cell cycle. Now, where do we find this? Well, we mainly find it in large cells that are very metabolically active. I will talk today about plants, as I mentioned, but you can find it in many different species, also many different model species like fruit flies, mice, flatworms, and so on. But whether we find it in certain cell types or not depends both on the species, but also on the cell type. And you can see some examples here in the table on the right. So in Drosophila fruit flies, for example, we can see that the ovary cells can reach up to 32C, But then there are certain cells in the salivary glands which can reach fluidity levels up to 2048 C. So that's super high. So they have a very high DNA content. And then Arabidopsis, which is my favorite model organism, of course. It's the little plant here that you can see at the bottom. There, the pavement cells, so those are puzzle-shaped cells that you can find in the leaves. They reach fluidity levels up to 16 C. But then the leaf trichomes, which are small hairs on the leaves, they can reach normally fluidity levels up to 32 C, but in some mutants, they can also reach 64 C. So in plants, endoreplication is involved in their normal growth and development. So even under non-stress conditions, endoreplication takes place in plants. And it's linked to cell differentiation. So when cells get a specific function, so they differentiate, that's when we see endoreplication occurring. And what's very interesting, this is not true for all cell types, but for many cell types, we see that there is a link between the nuclear DNA content, so the number of endocycles that the cell has gone through, and the cell size. So generally, cells with a higher DNA content are also larger. And some interesting examples of this are, for example, the trichomes that I just mentioned. So you can see them very nicely on this left image here. So you can see these tiny hairs on the leaf surface. And I call them tiny now, but actually they're quite big because you can just see them with the naked eye, but they only consist of one cell. So the cell actually gets very big through endoreplication. Another example of a tissue in plant cells where endoreplication takes place is in the endosperm. So this is here some sort of food reserve that is stored within the seed to make the plant embryo grow. But endoreplication is also often induced in plants under stress conditions. Of course, plants experience many different stress conditions. They're sessile, as we call them, so they're stuck in one place. So whenever it gets too hot or there is too much water, too little water, they cannot just run away from these stress conditions because they're fixed in one place. So there are many different stress conditions that they experience, as you can see over here. And what we often see is that then endoreplication is increased, so the DNA content increases. Now, researchers are actually not sure why this is the case or what the function is of this increase in endoreplication, but there are different hypotheses of why this might happen. For example, the induction of endoreplication could prevent the transmission of damaged DNA, because if cells would go through the classical cell cycle, they would double their DNA, which is already damaged, and then they would split it over two daughter cells. But with endoreplication, they would still double it, but it would not be split over daughter cells. Or another hypothesis is that this increase in the amount of DNA could also increase the amount of DNA that's available as a template for transcription. And then we could think, for example, about the transcription of genes that are very important in defense responses, such as, for example, antioxidants, which we will also still talk about later this week. But there's also many other hypotheses. So at this point, it's not yet clear. Now, of course, we are talking about flow cytometry today. So now the question is, how can you now analyze endoreplication using this technique? And here you can see a brief overview of what we typically do in the lab. So we measure endoreplication generally in individual leaves of our Arabidopsis plants. I will show you later why. And what we first need to do is extract the nuclei. So compared to animal cells, plant cells have very rigid cell walls. So we need to get the nuclei out of the cells. And we do this simply by chopping this leaf with a sharp razor blade and a certain extraction buffer. Then subsequently, we will filter this extract. As Carla already mentioned, this is important because otherwise you will just get clogging of the machine, which of course you want to avoid. And then we will stain our nuclei with propydium iodide. This is a stain that stains DNA. So of course, when we want to look at the DNA level, we want to stain the DNA. And then we will perform flow cytometry, and this is the device we have in our lab at Hasselt University. Then what will happen inside the flow cytometer? Carla already nicely explained this. Actually, it's exactly the same figure. So you will have the sheet fluid here, the sample is introduced into the sheet fluid, and then through this hydrodynamic focusing, you get really these individual particles, which will then be excited by a laser. Because we're using rubidium iodide, we need to use a laser with an appropriate wavelength for excitation of PI, and this is a blue laser with a wavelength of 488 nanometers. And then we measure, something got lost here, but we measure, first of all, the forward scatter, which tells us something about the size of the particles. And we also measure the fluorescence intensity of rubidium iodide. And this we do at around 580 nanometers. So we use a detector appropriate for that. Now, how do we then proceed with the data analysis? So first of all, we will obtain the so-called dot plot. And here, what we see on the x-axis is the fluorescence intensity of rubidium iodide. So this is a specific detector that we use for that. And on the y-axis, we see the forward scatter. And you will already see you have a lot of dots here in this graph, and each dot represents one particle measured by the device. You will see that here you really have very many dots very close together, but these are actually not the nuclei. These are not particles that we're interested in. This is just some debris. What you also need to take into account in plant cells, you have many autofluorescent compounds, such as chlorophyll, for example, and these can also interfere with your measurements. So everything that we see over here, we're actually not interested in. However, what's very nice is that over here, I hope you can all nicely see that, we have 4 or even 5 of these clusters. You see these thicker stripes, let's say, and these actually represent our nuclei. And each of these clusters represents a group of nuclei corresponding to one specific fluidity level, so one amount of DNA. So we know that these are our nuclei. So because of that, we put a gate around them. And then in our data analysis software, we can say that in this next graph, this histogram on the right, we only want to see the particles that are inside this gate. So all this rubbish, let's say, on the left side, it's just left out. So now we focus on these. And then here in this histogram, again, on the x-axis, we have the fluorescence intensity of rubidium iodide, so the same as in the left graph. But now on the y-axis, we have the count, so the number of particles, in this case nuclei, corresponding to each of these fluorescence intensities. And what you will see then is that you get 5 nice peaks. The first one corresponds to cells which haven't undergone endoreplication, so they have a DNA content of 2C. The next one are 4C cells, then we have 8C, 16C, and 32C. And what we can then do, I don't know if it's super clear here, but we can put also these small markers around each peak, and we can make the data analysis software count how many particles or how many nuclei correspond to each peak. And then we convert these values to percentages. As you can also see, here we used a log scale on both axes. The reason is because, of course, here our 2C nuclei will have a certain amount of DNA, so a certain amount of rubidium iodide fluorescence. But the 4C cells, or the 4C nuclei, have twice the amount of DNA, so we expect twice the amount of fluorescence. Then for the 8C, it's again twice the amount that we find in the 4C cells, and so on. So if you would put this on a linear scale, each next cluster would be twice the distance from the previous one. So it would be really spread out, and that's why we put them on a logarithmic scale. Another nice thing to see here is that these clusters, they go slightly up. So you see you have this nice diagonal. And the reason for this is, as I mentioned, cells and also nuclei with a higher DNA content, they're generally bigger. And that's also what we see here, because remember, the forward scatter tells you something about the size of the particle. So here you can see indeed a higher DNA content corresponds to larger nuclei. So as I mentioned, endoreplication is important in plant development. So during my PhD, I also managed to nicely show this in Arabidopsis. So what I did in my research is I grew Arabidopsis plants. We typically do this in a hydroponic cultivation system. And at some point, the leaf rosettes, as we call them, will look like this. And you will see here that I numbered the different leaves. So how an Arabidopsis rosette grows is at first, these two leaves, number 1 and 2, they will appear simultaneously. And afterwards, each of the subsequent leaves, so 3, 4, 5, 6, and so on, they emerge one by one. So here, leaves 1 and 2 are the oldest leaves, and then all the other ones are younger. What I did then is I performed this flow cytometry to determine the nuclear DNA content on leaves 1 and 2 of these plants, but at different time points after sowing them. And here you can see the size of the rosette. So after at 11 days after sowing, they're still quite small. After 21 days, they're of course much bigger. And then I checked the nuclear DNA content over time in this first leaf pair. And here you can see the percentages of cells corresponding to each ploidy level. So you can see that initially we mainly have 2 and 4C, a little bit of 8C, but then as the plants and also these leaves get older, you see that the percentages of these lower ploidy levels start decreasing, and you get more and more of the cells with higher DNA content. We even reach up to 32C here. So here on the graphs on the right, it's actually the same thing, but now I just plotted each of these different colors in an individual graph. So here it's also nice. You can see that immediately from day 11 onwards, 2C DNA content starts to decrease. The 4C peaks around day 12 and then decreases. The 8C peaks a little bit later, and so on. So we really see that as the leaves grow older, the cells have gone through more and more endocycles. Now there is also another parameter that we calculate, which is the endoreplication index. And this is the average number of endocycles that has taken place per cell. So how do we calculate this? So we just base ourselves on these percentages here. And we multiply each percentage by the number of endocycles that is needed to reach that ploidy level. So for 2C, that's 0, because a 2C cell has not undergone endoreplication. For 4C, you multiply with 1, 8C with 2, 16C with 3, and 32C with 4. Then you add up all these values. And I forgot to add here that you still need to divide by 100. And then you get, as a result, a value somewhere between 0 and 4. 0 would mean that none of the cells have undergone endoreplication. 4 would mean that all cells have undergone endoreplication. And this is just nice because it gives you one single value that you can compare between all of your samples. So in this case, this is the same graph that I just showed you. And now when we calculate this endoreplication index, which tells you how many endocycles an average cell has gone through, you'll also see that it increases over time. Then I did the same thing, but instead of following one leaf or one leaf pair over time, I waited until my rosette was 21 days old, and then I analyzed ploidy levels into each of these individual leaves. And here you can see on the right we have leaves 1 and 2, which are circled here, so the oldest leaves of the rosette. And on the left, we have leaf 8, which is the youngest one that I analyzed. And then you see exactly the same pattern. So again, with increasing leaf age, you get increasing ploidy levels. So here you again see the same thing. And when we then calculate the endoreplication index, we could nicely see again that with increasing leaf age, also the nuclear DNA content increases. And this is quite nice because it indeed means that throughout development, you get more endoreplication. And also it means that instead of harvesting the same leaf age or the same leaf position over time, it's actually much more convenient for us as researchers. We want to save time, of course. Then it's much easier if you just harvest different leaf positions of one rosette at one time point, because then you also know that you have different leaf ages that are corresponding to your samples. And then I also mentioned that endoreplication is also involved in plant stress responses. So this is something that I also studied during my PhD, and I specifically worked with cadmium stress. So as I mentioned, we typically grow our plants in hydroponics. So this is the system here. We have these black boxes, which are filled with a nutrient solution, with everything that a plant requires to grow. And then here on top of this box, we have these tubes filled with sand, and that's where our plants are growing. Their roots grow into the box with the nutrient solution and take up nutrients in this way. And when we want to expose them to cadmium, for example, we simply add cadmium to our box with the solution. So a brief introduction on cadmium. So cadmium is a toxic metal. Unfortunately, many regions around the world are polluted with cadmium. And even though it's not essential for plants, they don't need it for anything because it's toxic. They still take up this cadmium because it looks a lot like other elements that a plant does need, like calcium, for example, or iron. And what will happen then is that you get an increased production of reactive oxygen species. Some of you might have already heard about this, but we'll also see this in more detail on Thursday. But this will then have effects on signal transduction because ROS are important signal transducing components, but these can also induce damage to the DNA. And DNA damage is something that is very well known to also affect cell cycle progression. So that's why I was very interested in checking how cadmium would now affect the cell cycle. So what I did is I grew my plants in hydroponics for 3 weeks under control conditions, and then I exposed them to 5 micromolars of cadmium. And then I took samples at different time points after the start of the cadmium exposure. One thing that I first monitored then was the rosette diameter, so the size of the leaf rosette. And here you can see this in black for the control samples and in gray for the cadmium exposed plants. So it's very clear that they're not growing very much anymore after you expose them to cadmium. But then I also checked the number of leaves in the rosette, and there I didn't see any differences. So the leaves are still emerging, but they're just staying very small. So I could conclude from this that cadmium inhibits rosette growth, but not leaf emergence. Then I wanted to look at these different cell cycle parameters. So I took samples after 3 days of cadmium exposure, so relatively short-term exposure. First, I then looked at the surface area, so the size of the different leaves. So again, I have the oldest leaves here at the right, youngest leaves at the left. And then you can see very clearly that the size of all the leaves is much smaller if they have been exposed to cadmium. Of course, the size of the leaf could very strongly be a consequence of effects on the cell cycle. So in order for a leaf to grow, this can happen in multiple ways, but you can have differences in cell division because, of course, a smaller leaf might contain less cells, or it can contain smaller cells. And the size of the cells, remember that this is likely related to endoreplication. So to check whether I had differences in cell number or differences in cell size, I first performed a differential interference contrast microscopy. And here, I drew some cells in these black lines to make them more clear because I don't think the image is super clear. But what you can see here is the pavement cells, so these puzzle-shaped cells inside the leaf. And then I measured and counted the number of cells inside the different leaves. And I obtained the following results after 3 days of cadmium exposure. So again, in white, you have the control bars, in gray, the cadmium exposed plants. And you see that in general, the cells are a little bit smaller in the cadmium exposed plants. For leaf 5, this was significant. For the other ones, not, but you could see more or less the same trend. Then using flow cytometry, I also determined the endoreplication index because this could be linked to cell size. And here indeed, I also saw a similar trend, so slightly lower values after cadmium exposure, which was significant in the oldest leaf pair. So we could see that cadmium indeed inhibits both cell growth and endoreplication in the epidermal cells, so these puzzle-shaped cells. Then another thing that I did is I looked at cell number. So this will tell us something about cell division because, of course, to have a higher cell number, cells need to divide. And here I could also see some significant effects, significant in some of the leaves, not in others, but again, a general downward trend. And at that point, I got very annoyed by the fact I had to do all this microscopy because it's super interesting. It gives you very nice data, but it's also really a lot of work because in these images that I showed here, you really have to measure each individual cell. You have to count all the cells, so it's really a lot of work. But then I thought, I'm doing this flow cytometry anyway. Can I also not use my flow cytometry data to somehow also assess effects on cell division? And then I thought, yeah, as I showed you, you have to chop your leaves in a nuclei extraction buffer. And then I thought maybe the number of nuclei that I have per microliter of flow cytometry extract can also tell me something about the number of cells I have in each leaf. And this is something I tested. So here I also saw these negative effects of cadmium exposure, and they looked quite similar to the effects that I saw on cell number, which I determined really using the microscopy. And what I then did was for each of these data points, I put them on this type of graph to check whether there was any correlation. And then I nicely saw that the cell number, which I determined really via microscopy, was very strongly correlated to this new parameter, so the number of nuclei per microliter, which I determined using flow cytometry. So you can see here that the correlation was very strong. So the conclusion from this was that the concentration of nuclei in flow cytometry extracts can actually be used as a proxy to assess effects of stress conditions on the extent of cell division. Still, doing the microscopy is still the golden standard. Then you really visualize the cells. You can see how many cells there are. You can see their size. But we're doing the flow cytometry anyway to check on endoreplication. So this is a very nice additional parameter that we can include. So these were the results after 3 days of cadmium exposure. Then the last thing I was wondering about is if we expose the plants for longer, do we see stronger effects? And that's why I also took samples after 12 days, so almost 2 weeks of cadmium exposure. Again, here I no longer included the 2 oldest leaves because they were already senescing, so they're really aging and then they're turning brown and very dry, so you cannot really use them for analysis anymore. But I used leaves 3 up to 6. Here again, I saw that the size of the leaves was much smaller after cadmium exposure. Then when I looked at endoreplication measured via flow cytometry, now I saw significant decreases in the endoreplication index in each of the leaves I analyzed. Then when I determined this new parameter, the number of nuclei per microliter, I also saw significant effects on each of the leaves analyzed, except for the 3rd leaf. So I could conclude that also long-term cadmium exposure inhibits both endoreplication and cell division, so both variants of the cell cycle. So to conclude, I saw that cadmium exposure disturbs leaf growth and development, and that this is a result of inhibition of cell division, but also inhibition of endoreplication. So both variants of the cell cycle are affected. And I also saw that the effects increased over time, because after 12 days of exposure, the effects were much more pronounced than after 3 days of exposure. So a few take-home messages on endoreplication. So we saw that it is an alternative version of the classical cell cycle. It consists of DNA replication without intervening mitosis. It's involved in normal plant growth and development, but it's also very strongly affected by environmental stress conditions, such as, for example, cadmium stress. And the most important thing for today, I think, is that you can assess it via flow cytometry.